Ready? Okay, here we are. So for yes, some acknowledgements. For some of the slides that I will use today, I'm using the material given by Lehman and this Frankel Pinter. So they are scientists working for the study of the origins of life. And so they both presented to the last prebiotic chemistry summit, the PC3, organized by NASA. And if you want, I can give you the complete presentation of them. And also remember to you that if you are really interested in the topic or you wish to search something related to the prebiotic chemistry in order to make your presentation, you can search the PC3 YouTube channel, and you will see you have the direct access of all the presentations that were made the last November. Okay, so I'm just using some of the materials, so I wish to acknowledge them, and then I made some modifications. So I mixed also the slides. So we spoke about the building blocks. We divided more or less into 4 categories. So the proteins, carbohydrates, nucleic acids, and lipids. At the moment, as you know, we never spoke which is the most important, which came first. Also, you heard from Mansi that nowadays the prebiotic chemists, they're not thinking that one of them raised it before, but they thought that maybe they appear together more or less at the same time. As you know, the amino acids are the first one to be synthesized by a prebiotic chemistry approach. And then also carbohydrates for the formaldehyde, nucleic acid from hydrogen cyanide, and lipids from glycerol and fatty acids. So nowadays, these are the polymers, and all the reaction, the formation of the bigger molecules, they are reaction that gives you some water. So water is released when you create the links in between these monomers. So in from the chemistry point of view, this is called the condensation. So you have the condensation when you have the release of one simple molecule like water or other simple molecules. So the strength thing of this is that the monomers and they are water soluble. So that liquid water is essential for the origin of life because you need some mixing. But in any case, the reaction that gives you water as a product, they are not like to react in water because you have the condensation of A plus B giving AB plus water. But usually this is an equilibrium reaction, so it tends to go back. So if you work in water, usually you have an excess of water because the 1 liter of water is 55 molar. So moles on 1 liter, while the concentration of this maybe is millimolar if you are in concentrated environment. So it means that usually in water, the gas reaction goes in the other direction. The reaction does not go from the reactant to product. So you need something to remove the water. And this could be straightforward to find it. So the simplest way, just following the Darwin's idea. So the Darwin said, okay, so we have, imagine one pool of water. You have water and the compounds. The sun will hit the water. You have the drying of this. So by removing the water, heating a little bit, you have the loss of water. Then after some time, so you will have rain. So again, you will have the pond rich of water, and again, you can create a cycle. In this cycle, you can have the condensation of molecules. You can do it easily, for sure, in the laboratory. But when you do this, the reaction is quite a mess. What it means, you can produce some oligomers, but so you start, for example, here are the condition. You have to heat a little bit. For example, for the glycine, so the simplest amino acid, you have to heat up to 130 degrees for 1 day. If not, you have no reaction. For the glucose is just more delicate. You have to heat 50 degrees for 7 days, so more time and lower temperature. You can produce some oligomers up to 10, 12 monomers, but the problem is so many references. These you can find even these are recent, but you can find also older. Because from the beginning, the prebiotic scientists try to take the amino acids to produce the proteins. The problem, which is that when you repeat the cycle, at the end you will obtain some tar or char. So at the end, you will, if you continue to do this, to cycle this molecule, at the end you will have only more or less carbon. And what the scientists call the toluene. So toluene, Yuri invented this name. Toluene is the name of some carbon-containing compounds, so a mixture of carbon-containing compounds, more or less red-colored, like that you can find on Jupiter, for example, on other planets. So we know that they're rich in carbon, but we don't know exactly the composition. In any case, this it's complete a mess because you have some oligomers, but still the mixture is complex. Then you can have also some branched structure. So usually you like to have a polymer like this, but you have some branches, and also you have some the composition of amino acids, for example, sugars. And also you cannot control the polymerization. So sometimes the polymerization occurs in, for example, imagine from the DNA, you need to attach the phosphate in the correct position of the OH group. If you start with nucleosides, you cannot control the reaction. So you have all the isomers, possibly isomers together. Also, there is a big problem of this approach because when you dry everything, even you will lose the role of the membranes. So, nowadays, so the prebiotic scientists think that the membranes, so lipids and the membranes are very important for the compartmentalization, so the division of the reaction in the cell, like in the actual cells. And if you dry everything, you will destroy the micelles and everything. So at the end, maybe you are creating too much entropy. So because at the end you are mixing the food and the energy. So maybe this could be the preliminary step, but even not so well known. And so then if you cannot use only heat, if you see from the beginning, so here the timeline from the 60s, so the birth of the prebiotic chemistry to nowadays, many scientists thought about to find some prebiotic molecule able to promote the polymerization. You see, we started from the cyanamide. If you remember at the beginning, scientists likes to work with cyanate derivatives because they found into the external into the space. And also then you have the time for the phosphates. So like some of you, they think that the phosphate were important because nowadays you can find phosphate everywhere. So if you remember when we spoke about the phosphate problem, the phosphate is not soluble, but one cyclic trimer of the phosphate is more or less quite soluble. This is the trimetaphosphate. Also, it's very cheap. You can buy this for a few euros per kilogram. And so if you use it, it can produce some polymerization. If you see here, it is represented the yields of with respect to the peptides, so the number of monomers that you can able to attach. If you see the 3-metaphosphate, not so bad. And then, if you remember, because we are also the prebiotic scientists are also following the fashion. So at the beginning, there was the fashion of the hydrogen cyanide, then the fashion of the phosphate. Then you have the fashion of, if you remember, the hydrothermal condition. So hydrothermal condition that you use some sulfur-related chemistry. And so they use the carbonyl sulfide, and maybe they use some high temperatures and pressure to make this. So they obtain something. Then the last period, if you remember also from Krishnamurti, he worked on some diamide phosphate. And so everybody of these scientists for sure in their paper write that they think it is truly prebiotic. So at the end, what we have up to now, so we have some conditions, we have some formation of polymers, oligomers, more or less. The problem is always the same, that you create some oligomers in an uncontrolled way. Even these polymers are very short in length. So if they are short, it's difficult to find some property of them, because if in order to have a catalyst, maybe you need more. And so they are too simple to drive some chemistry. Also, you have some side reactions. So, for example, you can find some cyclization products. Or at the end, why you stop? Because for the reduced solubility. So, if you neglect the nucleic acids, so nucleic acids are very soluble, because the nucleic acids contains phosphates, though, so you have some negative charge, and so they are ionic, so they are soluble. For all the other compounds, it's not like this. So if you have polyamino acids, so the proteins at the end, if you increase the length, they will become less soluble. More or less the same for the poly sugars, because the poly sugar that nowadays we have is the starch that is quite soluble, that is branched, and you have some cellulose that's completely non-soluble. So, but at the end, when you increase the size of the molecule, you will reduce the solubility. Or you can have some side reactions, some hydrolysis of the product. And so I'm seeing someone new is writing. Yes. Yes, also this is a matter of debate, because then we will see later. So before, you need to promote the polymerization. So I never told you, but every one of these scientists, they choose the best condition for them. So for example, for the carbonyl sulfide, you use some acid chemistry. For the when you use some cyanides, you don't want to use the acid chemistry, because you will produce hydrocyanic acid. This is too much toxic. So maybe you work in alkaline condition. Also for this trimetaphosphate, you work in a condition that you are able to keep the trimetaphosphate in solution. So all these scientists use a different pH, different temperature. But at the end, the problem is that in order to have some system that is able to live, then we'll see later, you need to make a polymerization, but also you need to make also some depolymerization, because you must have some recycling of the structure. If you continue to use, for example, the heat to produce the polymers, at the end, the recyclability of these compounds is very poor, because you have some degradation. So if you degrade, for example, the side chains of the molecules, you cannot have the good recycling of the monomers for sure. So, but so we saw the drawbacks, but sometimes these are not complete drawbacks, because for example, we spoke about the cyclization, so the formation maybe of cyclic oligomers, but sometimes for some reagents, for some reaction mechanism, they take advantage of this cyclization. Also, we saw, if you remember, some scientists use the cyclic phosphate. So this the isomer, the cyclic isomer of the phosphate. So for this, if you remember, I spoke about the polymers of amino acids. If you include some polar side chains or use other solvents or temperature pressure, maybe you are able to keep in solution. So for this, you can also, it's not written here, but you're right, also we can work on the pH, for example, to keep the polymers safe. And so in any case, so many possibilities, not completely drawback, but something hindering the process of polymerization. And then another kind of scientist, for example, this Ferris. Ferris he thought that some minerals can promote the oligomerization better than the solutions. And so he's worked with the clays, so more nice, for example, but also other clays. I don't know if you know, but the clays on the Earth, they are new. They are just quite new because they are not formed directly from the volcanic eruption, but from the interaction of some special, like sand compounds with the water. Even if you remember, if you are following, you know, nowadays on Mars we have some famous rovers like Curiosity, for example. So Curiosity found some clays on the surface of Mars. So this is indirect evidence that on Mars you had water some millions years ago, because the only way to for the formation of clays that are rich in OH is the presence of water. So we know from the mineral chemistry on the Earth. And so if you are able to produce, so use the Mount Moralonite, so use other clays or minerals, you can have you can help the formation of oligomers, especially from the nucleobases. You see, they're using a trick, so not using heat, they're using this compound. This is activated phosphate for the oligomerization. And this is true, so you can do this in the laboratory. Then the problem is to remove the the compounds from the clay. This is another problem. So, but in any case, some other scientists are even they thought even that the clays and the minerals played a bigger role at the beginning of life, and so some of the theories are related to some template polymerization. So the RNA is able to locate some ions on the surface of the clay, and this helped the like the transfer of a reaction and the duplication of RNA. But these are just theories. So this is the truth. So you can do this in the lab. It's so simple to do this. Then it's difficult to remove from the clay. In any case, so we are, okay, it's working. So with many simulation, you can show that it's possible, at least from the basic principle, to induce some template direct synthesis. Here we see from like a sample of some RNA. We know that the template can drive the polymerization, but always we are thinking, because you think about the biology, so biology is able to duplicate RNA and from RNA to DNA to copy the information. And then when we go to the protein, we have only one sense. So the protein cannot be like transformed directly from the biology into the nucleobases. So we thought, but this idea of RNA world, that you need for sure some nucleobases to make the base pairing to make the template directed synthesis. But this is not true. This is not the truth. Because we know from the chemistry that you don't need always to have the nucleobases. Sorry. But in a laboratory, you can do something from like simpler science with respect to treating RNA and DNA. So imagine to have a substrate. This is like colored, like this one. So you have a substrate, and so you use some monomers like this one that can make some non-covalent interaction with some part of your substrate. So this is your substrate, and these are monomers. And you see this is it's not just for fashion, but I wrote this because it's a double bond. Usually, as a monomers, we use acrylates because chemists like a lot of acrylates. They can produce acrylates ending from with many possible groups. So this group can have some weak interaction with the monomer. So at the beginning, you have some template of your substrate. So the monomers attached to the substrate, for sure, they are in equilibrium, so they can attach and detach quickly from the solution. And then after some time, you can start the polymerization. You initiate the polymerization, you can use the light, you can use heat with respect. So this usually, when you perform in the laboratory, is a radical polymerization. And this, when you make the polymerization, you are fixing the position of your recognizing monomers. And then if you are able to remove the substrate, you will have a hole, you will have a cavity able to take back the substrate. And so the technique here is used in many cases for the analytical chemistry, usually, but it can be extended to other interesting fields. And it's possible also to create some catalysts, so enzyme like imprinted polymers. So as you are seeing here, we are using like monomers. They are for sure not prebiotic, but what I'm telling you, because at the beginning I told you that for the chemical point of view, you don't need exactly to have RNA or the amino acid. You need only to have some monomers that have suitable differentiation, so they can make some non-covalent interaction, and then you can induce the polymerization. So I'm not telling that acrylates are prebiotic safe, but I'm telling you that this approach is like you can use for the different polymer that you want. So for example, you could create some molecular imprinted polymer starting from, for example, amino acids. And so the idea is exactly the same. So at the end, you can create also enzymes. So you can create some compounds that are able to make some catalysis. How they do this, if you want, you can read the paper, but it's very technical. But they imagine, you remember the theory of the reaction. When you have the reaction, so this is the energy. If the reactant goes to the product, it's because the product has less energy. So for all systems that you want, that you are considering, the system evolved to have the lower energy. But in order to do that, sometimes you have to reach to higher energy. So you need to go to some transition state. So you need that your compounds make some arrangement, they in some shape, and they give you the possibility to go from the reaction to the product. So with the catalyst, you will lower this. But in any case, you still have some transition state. So transition state is something that you like living for a few nanoseconds or picoseconds. So it's not a molecule. It's like molecules reacting together. But if you are able to imagine the transition state that is similar to a molecule, so with a shape, if you create a molecule, then you create a molecular imprinted polymer. You remove the molecule, you remove the template. So you are creating one molecular imprinted polymer that is able to make the catalysis, because inside here you will have the transition state of the reaction that you are working on. So it's possible to do this. Even the molecular imprinted polymers can work in different scales. So they can work from molecules. So monomers, so like small molecules, but even peptides or big protein or lipids or some complex protein. So protein with some glycoside inside, or even bigger objects like virus, bacteria, or eukaryotic cell. So the molecular imprinted polymers, they can work at these different scales, as you can see from the nanometers to microns, more or less. So we use this. Sometimes they are used also for the cancer treatment. So this also you can use. You can create some artificially antibodies. So they call these plastic antibodies. And for this is an example because you can find many in the literature. So this example in which you take one nanoparticle. It's not silicon, it's silicon dioxide, but the authors write silicon. So it's like a silicon dioxide nanometer size bead, and you cover this with the molecularly printed polymer. So you create the polymers on the surface of this nano-sized silica. Then you remove the template, and this silica particle is able to attach to the sialic acid that you can find on the surface of some cancer cells. And so you can induce some attachment of this particle only on the cancer cells. So you see, we are using something that is like plastic because we are just making usually these are with techniques use are acrylates. Acrylates is like the plexiglass. You know what is the plexiglass, what I'm speaking. So like plastic, but we are imprinted. So in nano principle, if you substitute the monomers with some other monomers, you use a different chemistry, you can do the same. So from the chemical point of view, if you use one monomers or the other, if they are sufficiently different, so you can do whatever you want. Also, from the prebiotic chemistry, so we are always concerning that, okay, we take amino acid, we must need a peptide. But we have many differences. So if you, for example, if I take this water bottle is not made by the polypeptides, but it's made from a reaction similar to the second one. So this bottle is a polyester. So the condensation of some acid with an alcohol. So we are using polyesters every day, but polyester can also have been present in the prebiotic soup because from the reaction, for example, of Miller, you have the formation of this hydroxylic acid. So this the hydroxyl, and this is the acid group. So they can make some reaction of condensation. So again, the removal of water. So you see here the part. So the molecule can make a condensation with another molecule of hydroxylic acid, and you can produce some polyester. So nowadays the polyester are more or less, not always, more or less like plastic that we use every day, as I told you, the PT, other polyester that we have on the market. But remember that if you go in the on the vegetables, the cutin and other surface, some plants have one natural polyester coating that is able to avoid the loss of water. And so many, if you see some plants, maybe they are very smooth on the surface. If you touch them, it remembers sometimes the plastic, and this is true. So some vegetal cells are able to produce some polyesters on the surface. So completely polyester will be more complicated in this one, but they can do it. But also you can have a mixed chemistry. You can have something that is sometimes like a peptide, because the peptide is characterized by this group. The polyester is characterized by this one, and you can have some something that is in the half. So for some chemicals it's polyester, some others it's a peptide. So it's called depsipeptide. Also, depsipeptides are known into the actual living organism. Okay, and so depsipeptides are very well studied, maybe in the last years, last 5 years, more or less, of the prebiotic chemistry, because nowadays the approach of the prebiotic chemistry is to use everything. So not just to select the amino acid that you want, but to use a mixture of hydroxy acids and amino acids, and you can find some depsipeptide. It's easier, so they are easy to find. They're easy to be formed, and also you can have some exchange in between the ester bonds and peptide bond. So it's possible to transform this step by step to a deoxypeptide and slowly to this one. So it's like you are increasing the reactivity of the bond that you have here. Okay, so this is a mixed approach. So this backbone heterogeneity, for sure, it seems that is a mess, but for the reason that I told you before, so maybe this is not. So it could be a step, it could be maybe not, was a step, but even let us to think more related to the what we call today the system chemistry with respect to the normal chemistry approach. Then, for example, if you think about some like ribozyme, still you need for the smaller that you can find, so 86 unit, they are maybe too complex to produce directly. But there are well known many examples in which you can split down the structure in a smaller structure that in line of principle you are able to produce from the prebiotic point of view, because if you remember for the oligomerization that I showed you for the RNA with the clays, you can go up to 40 units, so 31st is not so different. And you see these units, even if they are smaller, they can make some template arrangement to produce a structure that is it has more or less the same activity of this enzyme. So maybe for the prebiotic enzymes, you have the enzymes that are able to be produced for smaller polymers. So maybe they don't need some bigger chain for the production, because I have to remember you that the self-assembly from the molecule point of view is very, very important. If you remember, for lipids is they are very easy to assemble. So you produce a molecule. The molecule could have, for example, the shape of one cone or one cylinder or other shapes. And according to the shape, according to the charge that they have, they could arrange in the micelles or create some double layers and so on. So this just normally. So when you synthesize the molecule, they make the assembly related to the concentration. So high concentration they assemble, lower concentration they deassemble. So this is called some micellar concentration. And for some polymers, they can do the same. Even if they haven't the nucleobases, they can make some self-assembling. They can produce some fibers or nanotubes or other different structures. So don't think that you need very long polymers in order to create some fibers, even from the macroscopic point of view. So we spoke about the polyesters, and so people working even on like polyester microdroplets, thinking that this could be related to the compartments needed for the origins of life. And so you can produce this polyester in a solution. It's with a easy chemistry. And what is known, why this is interesting from the prebiotic point of view, is because these like small bubbles, they are able to make the concentration of some interesting polymers from that are biological polymers. So they concentrate, so if you have RNA in solution, you have these droplets of like plastic, more or less droplets. The RNA concentrate inside them. So the concentration inside these droplets, it's higher with respect to a sterile one. So again, we even if you don't think that the lipid world came first, we are seeing many times that we need membranes for the safer environment of the cell to concentrate also the compounds to create maybe a metabolism and so on. Also, you can obtain the same in prebiotic condition. You can create some aggregates that they resemble like the amyloid structures. Using very simple monomers. Even another thing is that at the end, in order to create some enzyme, some molecular recognition, some self-assembly, you don't need too many amino acids. So nowadays, for the proteinogenic amino acids, you have more or less 20 amino acids. But in any case, it's possible to use less than 20. So some of them seems that they are very useful. Others could be substituted. And so you see the structure like the polyalanine, polyvaline. You are able to have the microfibers in solution. So they use only 2 of the proteinogenic amino acids. Okay, so they can form some quaternary structures, so they like bigger molecules. The molecules they are used, they are very small, but they can make some aggregation, for like this one. Okay, but so now I showed you it's possible to have some oligomers, but still we need to have, if you need to make some life cycle, we need to make something that nowadays is very well of concern. So you must have some recycling. So if you use the monomers, then at the end you have to get back to the monomer. So you have to remove the monomers from the polymer. So if you create the polymers, then you have to reduce. This is related also on the food. For example, we eat proteins, so we can decompose these to smaller units, and that we can build in another shape. So the life is also some recycling of the maximum possible of some compounds. Nowadays, there is not a clear idea on how to recycle the monomers from for example, from the proteins. If you are a biologist, if you need to break the links in between the amino acids, you must use some enzymes, some complex structure. So you have to digest the proteins, and maybe you have to use several enzymes to do that. If you wish to do this in the chemical point of view, you have to boil them with hydrochloric acid, so a strong acid, for many, many, many years. So at the end, you have the release of the monomers. But okay, this is difficult to think some prebiotic condition, because for the current biological approach, you need some enzymes. They are complex. If you use the acid, you have the very strong condition, not mild, as you can see in the cells. And so in order to have some simpler chemistry, so this guy invented some easily recyclable monomers, and he tried to use some chemistry that is orthogonal to the chemistry of, for example, polymerization. So he uses some sulfur-related chemistry. So the sulfur compounds, but even actually in the cysteine, we have the same. The sulfur compounds can produce some linkage like this one, and this is reversible. So you have giving from one side to other, you have to perform oxidation. Going back, you need to make a reduction. Maybe girls know this because on the hairs, on the structure of the hairs, they are made on proteins. These proteins have some cysteine inside, so you can change the shape from the hairs, so from curly hairs to straight hairs, because you perform some oxidation and reduction. So first you break these bonds, then you make the molecules free to move one to each other, then you change the shape, again you perform the reduction, goes back, create again the linkage in another position. So this like reversible. The condition that use for oxidation reduction, they are not so strong. It means that many molecules can survive to these conditions. And so if you choose the correct monomers, you are able to, with a click, to create the bonds, with other click to destroy the bonds. So you're able to make the recycling. So first you have the monomers, then you have the polymers. So what is interesting for this? Because now I talk to you about the approach of the template chemistry that you can have with the normal chemistry. In other principle, you can do, I told you, you can do the same with all the monomers that you want. So you can use also these monomers. And as you can see, you can choose from all synthetic way monomers to some monomers that, if you're able to see, they are made from some cysteine. So they are like tripeptides, for example. So this guy made a lot of examples, but also used these monomers that are like tripeptides. So if you are able, if you can use this template chemistry, you are creating one dynamic combinatorial libraries. It's something different from before, because before you made the polymer and then it finished. Now you can create, so you have a library of monomers. If they are sufficiently diverse, one from each other, you make like template chemistry. Then you choose which one is the best. And then you can make some improvement on this, and it's like driving this the chemical evolution. And with a click oxidation reduction, you can go to the formation of the bond or to the destroying of the bond. So with this, you can create some molecules, and for example, that are able to promote the self-replication. So they use the self-replication in the title, and so maybe self-replication is too much, but if you remember, I told you that for some approach for a chemist, if a molecule is able to make a catalysis of its own molecule, it's an idea of possible self-replication. So it started with one monomer. When the monomer aggregate to form some cyclic structure, the cyclic structure are able to template the monomers to create another cyclic structure, and so on and so on. Then this structure, you see, they are not covalent bonds in between the structure. So by shaking this, you can remove, so you are seeding this. And also you can stop them. So here you see this complex can be released because you have no bonds. But if you use UV light, you can mess up the bonds, creating some chemical bonds in between the shape. So this shape now is solid. Okay, this can be disaggregated, this not. And so you can, by changing this, also you can play with this, you can create some different self-replication, you can make some competition in between the molecules by using different templates, so different seeds. You can drive the formation of different structure. So at the moment, for sure, the structure are useless, and even from the prebiotic point of view, maybe not too much important, but they are important from the theoretical point of view, because they demonstrated that we are able to produce some self-replicating structure. We are able to make some recycling of the structure. So with this approach, with this dynamic combinatorial library, so we can do whatever we want, like related to molecular printed polymers, so to create receptors, to create some structure that are able to assemble together, or to create some bioligands, or some higher, so most complicated, like self-replication of the transport, because the transport is just a recognition of molecules on the top of another molecules having receptors. So with this dynamic combinatorial level, you are able to create some small chemical evolution into the test tube. and because you select only the one that is able to make a link on, for example, the target that you want. Okay, there are questions? Have you ever heard these dynamic combinatorial libraries? No?
Nope.
Okay, so just to, because today I told you we'll speak about polymers. So now we are neglecting a big problem that is on the basis of polymers. And what is this big problem? Is there some question? How these models differ from prions? Oh, more or less the idea is the same. So the prion is like a template that is able to produce himself. So from the point of view of the basic idea is the same. So the prion is a template directed systems. The prion directs the folding. So it stabilizes one form because it creates some template folding, and this is autocatalytic. So from the autocatalytic point of view, it's the same. So we found the easiest example that we found that I showed you is the glycolaldehyde, the smallest molecule that is autocatalytic. So it's autocatalytic in the presence of formaldehyde. And then, so these are autocatalytic, so they are like a bigger molecule. If you go on the prion is a protein, so it's very complex, but the idea is the same because the template directed polymerization or folding occurs at different scales. So from a small scale, so few atoms, to big proteins. That's the strength of this template directed synthesis. Oh, but so we never spoke about the chirality. And so nowadays we know when we speak about natural amino acids or some DNA, we know that the amino acids are like related to one form of amino acids. If you took, for example, the mirror image of the amino acid, you have two possibilities. The first one, your body is not able to use it. Second, it could be toxic. So when, for example, the simplest case, maybe chemists they know, if you have a carbon atom, because carbon make many bonds. So imagine to attach four different atoms or group of molecules, bigger parts. So four different objects to the carbon atom. If you make the mirror image, they cannot overlap one to each other. So the mirror image is another molecule. But for example, the nature chooses only one. So very few examples of maybe the different amino acids are found in the living system. Sometimes you can have, but very few examples. And this is a problem because not a problem, but imagine when you use drugs. So it means that this chirality drives the shape of one molecule. So for example, if I have an enzyme, if I'm able, but it's very difficult nowadays, to make the mirror image of the enzyme, so create the same sequence, but by using the mirror image of amino acids. So you have the enzymes working on the mirror image of the substrate. And so it means that drugs must interact with objects that are like called chiral in nature, because it's like hands, so they're different. So the left hand is not superimposable to the right. And so the drugs usually, if you want to make some interaction with the enzyme, protein, or something else, the receptor, at the end, almost surely you must have something, you have to deal with the chirality. It means that the mirror image of the drug is not active, or maybe it's very toxic. That's why the organic chemistry boils down on the preparation of stereoselective compounds and anti-selective reaction and so on. So because you need only to produce one form of be A or be not, but you must drive on the shape that you need, because the other part could be dangerous for the organism. It's very well known that in the past some drugs have was given, and there were two different forms. One is active, the other is very toxic. And so, but we are dealing with the polymer. So the chirality is related to atoms or some structure, but imagine now only now for atoms. We were speaking about monomers. So our monomers are chiral, only one type. So for example, for this one. And so if you attach them together, so I made an example with like a plastic material. This is a polypropylene. It's produced by humans, not natural. But the polypropylene, if you see this carbon atom is attached to 4 different ligands. And so if you're able to maintain the same position of this carbon atom, so the same chirality of this carbon atom, you're able to produce one compound that it has a shape resembling one spring, and this is very regular. And so if you are able to do this, you are creating one polymer that is called isotactic. And so if you know the isotactic polymers, so the polypropylene isotactic was invented by Ziegler and Natta. Because they invented one like catalyst able to form this polymer. So the isotactic polymer of polypropylene is very interesting because you are using for bows and other plastic parts that you are using nowadays in your life. But if you make a random polymerization, the polymer is messy, and so it has no shape, and it means that it's very bad for the mechanical properties. So the isotactic is very good in properties. Also, you can play with this one by the chemistry. You can make a inversion, so you can have some of this that is, you see they are alternated. So the methyl is one at the top, one at the other at the bottom. So this is called the symbiotic. And so also this is interesting from the point of view, not the random one. And for the nature is the same. So if you use the amino acids, if you are using non-chiral amino acids, so the amino acid that is a mixture of both left and right-handed amino acid, you will obtain a mess. But I never show you that in the middle experiment, you form one type of the other one. So in nano principle, you should form both. So now this is a big complication for the prebiotic point of view, because not only you have different compounds in the prebiotic mixture, but you could have also different shape. So the mirror image of the monomers. So we know now that if you use one shape or the other one, you are able to produce something interesting. But if you put this randomly, you cannot do anything. So we know from the polymerization that sometimes occurs. So imagine to start from the shape random monomers that could be present in both forms. When you induce the polymerization, the homochiral are maybe better preformed. When you have the random polymer, this is very bad in condition. So you have remove of part of the material. So at the end, you need something that is called symmetry breaking. So at one time, the nature must have selected one of these or the other one. So which one of these? Why I choose the right hand of left hand is there a reason for this? Only a chance. So for sure, we don't know exactly, but we have some ideas. So first of all, if you remember in the practice speech, he spoke about the role of the photochemistry in the prebiotic environment that is able to destroy the molecules, and maybe the actual nucleobases are the most stable from the photolytic point of view. And so if you use normal light, you will not obtain any inducing of chirality in your molecule. But if you use some kind of circularly polarized light, so the circularly polarized light, you can do it in the laboratory with some crystals. And it means that, so you know, when you have the wave, so electromagnetic wave, you have the magnetic and the electric field, they are perpendicular. Usually, for example, if you have a normal lamp, you will have photons that are in all the possible position. But you can induce some polarization. So for example, now I'm looking this screen. This screen is made with LCD technology. I have a polarizer. So I have a polarizer filter. Only one light is passing now. And I can see this with another polarizer. But if this screen is polarized linearly, so only photons that are, for example, the electric field in this position, they can pass. But if you use a circularly polarized light, it means that is the traveling wave. Imagine to make the rotation of this filter very quick. It's not the way. So you're able to produce circularly polarized light. And this light could be like left or right circularly polarized. And if you use one of these, you're able to induce some chirality in the molecules. So there are many examples. This is just to cite one. It's difficult to produce in the lab, but still you can do it. So you are able to create one or destroy also only one enantiomer. So enantiomer is the name of one mirror image. So with this circular light, you are able to make so destroy one of them. But so the sun is circularly polarized. Oh, not so much. So is there the possibility that we have some circularly polarized light in the universe? Yes, we have some idea. Some source, for example, in this paper, they saw that you can create like a big like jet of circularly polarized light from some gamma rays bursts. So in the universe, you can have some circularly polarized light. So maybe from this side of the universe, we have a little bit enhancement of one enantiomer with respect to the other one. So still is matter of debate, because if you remember, all the prebiotic chemists, they like to study the Murchison's meteorite. And the Murchison meteorite contains is the conductose carbonite. It contains amino acids. Some study they show that the amino acid chirality is little bit enhanced with respect to the amino acid that we have in nature. But the difference is very small, even it's a matter of debate. So we don't know exactly, maybe we have some contamination from the earth, so difficult to understand. But in any case, if we have this difference, is very slightly different from the beginning. So then what we know another fact for the atom that is called parity violation. This is a property of the nuclei of the atoms. This is Madame Wu. She made a crucial experiment in the 60s. And this experiment is, some atoms have a nuclei that is magnetic. So the nuclei of the cobalt-60 is magnetic. It means resemble a magnet. And so you are able to put in the magnetic field and make the orientation of these atoms. It's like in the NMR that you are using. So this Madame Wu made the same with the cobalt-60 atoms. And these cobalt-60 atoms are radioactive. So they emit some beta radiation, that is an electron. So she observed that the beta, so you have difference in between the amount of beta emitted from the top with respect to the beta emitted on the bottom. And this is due only to the possibility that in the nuclei, you have something that is chiral by nature. So starting from these experiments, the physicists measured the parity violation in many atoms, in many nuclei. Heavier is the atom, higher is the difference in the parity violation. And but if the parity violation is true, it means that the isomers, they have not the same energy. And so it's puzzled because the first year of chemistry, they teach you that the enantiomers have the same energy. So the left from the right-handed, they must have the same energy. But from the parity violation, this is not. So you have a contribution that is cannot be summed up from the mirror image. And so the one of these has a different energy. So, but how much is this? So we can calculate this. This is the calculation made in the 80s. The difference in energy is very, very, very small. So 10 to the minus 10 joule by mole. But they measure this with a big uncertainty, so 5 orders of magnitude. So, but we know we are measuring in atoms, in molecules, very difficult. So many people in the, actually, they made some calculation with some existing complexes using heavy metals, because higher is the atomic mass of the elements, and higher is the effect on this parity violation. So nowadays we know that enantiomers could have a different energy, but still we are not able to measure. It's a big deal because many scientists in the world trying to measure this parity violation, but you need very top of the art techniques. And also you need to synthesize some compounds like this one. You must have this in the pure form. So the measurement is very difficult. Up to now, nobody demonstrate that the molecules can have different in energy. So the enantiomers can have a different difference in energy that is measurable. But see the calculation. So from the theoretical point of view, if you made the calculation with some special techniques, so you must use the Dirac techniques. If you are able to make this theoretical calculation, you demonstrate that the actual amino acids are more stable with respect to their mirror images. So, but we must find this, we must measure. So the theory is not sufficient to saying this, maybe. And also the difference in energy is so small, how you can think that the difference in energy, or even if our amino acids are more stable with respect to the mirror image, maybe this is a chance because the difference is so small. So how it is possible? But in any case, even if the difference is slow, is very few, we know that there are some reaction able to make some chiral amplification. This is called the SOI reaction by the name of the inventor. This reaction is like a reduction reaction. So you make the chemical reduction of this carbonate to the OH group. And this OH group is chiral, so it can be also in the other position. So I don't want to write everything, but okay. So it can be up or down. And so if you perform this without a template, you will obtain a Racemi mixture as usual. But if you put like a slight excess of one chiral information, you're able to move the reaction toward one complete excess from the enantiomeric. And also you use it, for example, here the template is like alkane. So the alkane is almost No, because usually in chemistry, if I have some chiral induction, I must demonstrate that there is some interaction between the substrate A and the substrate B. But the alkane is very, very slow interaction, so very, very lower energy interaction with the B. But still, if you use you see the R or the S, you're able to drive the enantiomeric excess in one part or the other one. So you see complete enantiomeric excess. So by using just small amount of this chiral information. So the soil reaction, I don't go into detail with the mechanism, but the mechanism is like this. Why you have the current amplification? Because imagine that you have the complex giving you the, so first you start with the non-chiral complex, then you can have the R or the S. The problem is if you go only on R, only on S, you have a good amplification. But if you have both, you have a complex in between the R and S form of the catalyst, and this is stopping the reaction. So you are you're making a competition between the R and S because you are studying the difference in between the two. So for example, imagine to have the excess of R only of 0.1%. So the rest will mix together, exit from the reaction, and the 0.1% is able to drive the reaction. And so why it's working like this. So, okay, we stop here. So today I spoke to you about the polymerization reaction that you could have from the prebiotic point of view, even some template direct chemistry, and so the homochirality problem. That still we don't know because there are people thinking that the parity violation drive the selection of natural amino acids. Other people think they only want by chance that we have this, but we know that it's not possible. So at some time life must choose in between the left hand and the right handed. If not, the structure it's too mess. So you cannot have some good catalyst if you are mixing the R and S, so the two mirror images of the monomers. Okay, I think for today we can stop here. If you have any questions, comments. So today's lessons is like technical. Have you already heard about the homochirality problem? So as you can see, this is an additional problem that we have in the prebiotic science, because we know that we cannot deal with the mixed polymers having the two left and right image. But still we don't know if this happened at the beginning or at the late stage. So also I read some papers that because I told you in the beginning, some clays are able to drive some prebiotic chemistry reactions. And I know scientists that are using all the possible minerals they can find. And also some of them, they use the moon's regolite, so the material that you have on the moon's surface. So for driving some prebiotic chemistry reactions. Okay, guys, so if you have any questions.
I'll just add a curiosity. I'm looking at the molecularly imprinted polymers and the structures they can form. There's a similar theory when it comes to the production of cells, where porous rock forms a circular structure on which fatty acids can build up and generate cells. So this may just be theoretical, but they've actually shown that this can work for shaped productions of proteins.
Yes, for someone is like that because I think the name is Walter Schoser. It's the name of the scientist that thought like this. So we saw that because today I spoke you about the template chemistry by telling you only the polymer like organic polymers, but still you can have also some inorganic polymers. They're called geopolymers. In line of principle, so we cannot exclude that this material could have some driving the shape of molecules or cells or proteins. So for some of scientists, they are taking this very seriously. So they think that you can, so at the beginning, especially clays, they can have the shape like to print to create a lot of template directed synthesis. So you have the association, then polymerization, removal, and again, since the usually the inorganic polymer is strong, with one of the template you can produce some billion, trillion, or more molecules.
With the fact that enzymes are just shapes of proteins, I
Yes.
suppose you can have inorganic shapes at some point, which in a later point become, how can I say, they can auto-replicate with their structure with the DNA.
Yes.
Therefore, you're passing from an inorganic replicator that is just a shape to something that can synthesize DNA or RNA.
Yes, because we know that, so by chance or not, so that the RNA, DNA, they like to stay on the surface of these clays and other silicates. So they stick on the surface. They are sticky, and so for some of them, they have maybe the idea of Julia's idea. So that at the beginning there was, so why we have phosphorus in the DNA, because the DNA likes to be connected to some phosphorus containing minerals.
Mm-hmm.
So since they stick on the surface, the phosphorus was included, and then after that you have the
Selected.
yeah.
Okay, thank you.
Okay, thank you.
I have a curiosity actually
Yes.
regarding um, one of the points that we talked about today. I was thinking, but is there any way some traces of the molecules that were present in this period of time remain in the layers of in the lower layers of the Earth of our planet?
So that I wish to know if I understand clearly. So you are asking if
If there is a kind of fossil, I don't know, fossil molecules that remain in the earth, from that time.
so the most ancient stone that we have on the earth is more or less from 3 billion years ago. So we know that's why you have the big problem. If you think maybe more than 20 years ago, nobody knows. But nowadays we know that life on Earth comes quickly in the first billion of years ago, because the one of the most ancient stone that we found on the Earth, it is I don't remember if in New Zealand or in Australia, they saw that there is possible life on that one, because you see the fossils or some shapes of cells or group of cells. And what they found is exactly what you wish to do. So they try to analyze if you have still some molecules on the surface or inside that kind of very, very, very old stones. And so I read the paper. If I understand well, they didn't find anything. So unfortunately, the organic material didn't survive to the transformation of the cell, because if you think about these 3 billion years or more or less all the stones are melted and compressed or treated with water or the high temperature. Because also remember you have the Pangea in the past, so the first the land was connected, then disconnected, they connect again, they disconnected. So we have the tectonic. So our planet is quite has a very rich geology. So it's very difficult to find some stones that they are not heated or destroyed in some way for the mechanical point of view, or watering or something else. And so I think that this is even worse if you dig a hole. But so I wish to reply to you that Russians, for example, make say drill one hole like vertical hole, not for finding the oil, but just for checking the technology and checking what is on the surface, not on the surface of the planet, but on the deep crust of our planet, because we know a lot of things. Maybe from galaxies far, some billion years, light years from here, but we don't know anything about what we have under our feet. And so they dig this hole, this I don't remember, 3-4 kilometers, and what they found is they found some like bacteria, then bacteria that in the at this depth. So nobody saw that you can find some bacteria, but they found. So still you have some living organisms that are living under our feet. And so in order to reply to your question, I think it's very difficult to find one ancient rock that is not that has not been blown away by fire, by high temperature, by water, or by some organisms that are living under our feet at very high deep. But I haven't know. So all the papers that I read, they were not able to find any possible compounds.
I see. Thank you very much.
You're welcome. So I see one question from Paulo. I've not understood if in nature we do not see prebiotic chemistry because it does not exist, or because we don't know how to observe it. So both is true. The problem is if you think the first systems of the first prebiotic system, and if you think that nowadays the organisms they are selected, we can think that in the actual environment the primeval organisms cannot survive. Or imagine that we mix with we find one piece of dormant like primeval organisms. When you open this box, actual organisms will eat them, kill them, because they are stronger, maybe if you have some selection. But is it true also the second one? Because how, for example, I told you that the Russian digenol, they found some ancient bacteria. How can do it? It's simply in the laboratory, you take the sample, soil sample, you make some, extract the DNA, make some DNA amplification, they check it. But what, even if on the Earth we have some living organisms that are not living with the DNA, RNA. So for that case, we have no clues. So we don't know if we have that kind of organism without DNA or RNA. We are not able to see because we are not able to amplify these and to detect this. So, or the first two options. The first one, they are big, we can observe them directly. Second option, we are not able to saw them because we are not checking the correct chemistry. This is more or less the problem that we have on, for example, on other planets, for example, on Mars. On Mars now they are trying to find some maybe living organisms. So more or less they think that Mars everything is already died. But imagine that still you have some organisms, and we go there on Mars with a probe, and one of our bacteria or other microorganisms is on the probe, and he found useful to eat the other organisms, so we are destroying the other life. Or second possibility, we go on Mars, we found something that looks like the organism, and then we are not able to analyze it because we take it, even if we get back here on the Earth, we make some, what we do, DNA amplification, we don't see anything. You have one small imagine one small cell. How we can make a chemical analysis with the characterization? Is it not possible? So at the end, how we can analyze this? We have no idea, actually. So for some scientists, they think possibly even on the Earth, it could be. So we cannot exclude that some organisms here, they are without DNA, RNA. In that case, we are not able to see, to detect.
I just uploaded, there was an article about a week ago. They thought they had found some fungi on Mars.
Mm-hmm.
They were like these little spherical.
Ah, yes.
Yeah, so they proved it wasn't. So I just sent you a link.
Okay, okay. Yeah, it's called, they called Martian blueberries.
Yeah.
Yes, because also there was a matter of debate because replying to Julia. At the beginning, so they found some fossils, for example, these old stones, they found some similar to fossil. How you can know it's a fossil or maybe not? Just objects that are included, because it's not that everything in the millimeter, so maybe micrometer scale, they are fossil. Maybe it's like you created this like holes inside the stone by making some heat treatment, or we don't know exactly. But in any case, now we are almost sure because they found more and more evidence of this micro. The thing is so strange is that even for that organism 1 billion years ago, they resemble too much our actual organisms. That's the main problem. Because they look like bacteria. Also, they found a recent paper that makes some aggregation. So you have differentiations in between the cells inside. So you can find some group of these small organisms. So even we are adding more problems. So after 1 billion years after the creation of the Earth, you have the life. We have direct evidence with these fossils, but they are too complex. So it means that we are reducing the time for the life's origin. And this is a really big topic. So nobody knows exactly. So why it arose so fast?
I mean, the main problem, I think, it's creating a sort of context, because I don't think it's possible to get information about, I don't know, the right temperature, pH, or conditions in which the first molecules can be produced or were actually produced. And I don't know, but I don't think there is a way to date this bacteria that they found in the layer of the crust. So that's the main problem. And if I understood well, they don't have. I mean, they can have a DNA or maybe not. So it's. I mean, is there a way to timeline when they actually formed? No.
But you're speaking about the fossil that we found or other organisms? You're speaking about fossils found?
Yes, but even other yes, mostly regarding the fossil. I mean, they are fossil, okay. So we can place a time. We can place them in a right time.
Yes, we can do it. So we are not using carbon because it's useless. Carbon-14 is completely useless. First, because it depends on the activity of the sun. Secondly, because the carbon has a half-time of 5,700 years. So 1 billion years, you almost no carbon, so it's completely absent. But they studied other isotopes that are present on the rock. I'm not an expert in the field, but I can find the paper if you want. Because they made the calculation of the here I can see here. I'm reading down the cyanobacteria fossil record is among the oldest of any group of organisms, possibly reaching back to 3.5 million years ago. 3.5 thousand million years, so 3.5 billion years ago. So I'm just reading. I just found this paper. Not, I don't know if it the correct one. But they're speaking about cyanobacteria. So the strange thing, 3.5 million years ago, billion years ago, sorry, 3.5 billion years ago. So after 1 billion years after the creation of the Earth, we have cyanobacteria. So they're so complex. So nowadays, the cyanobacteria, they're complex. They have the membrane, the thylakoid membrane. They have.
They can do the photosynthesis.
Yes. So the photosynthesis is so complex. And you see here, they are phosphorites. So maybe you like this because they are minerals containing phosphorus. And I think they can.
Because I mean, just to contextualize what I was asking, there is this open question regarding what came first. So if it was the lipid world, if it was the amino acids or the sugars and so on. But the main problem is that you cannot place a time to whenever each of this compound arrays, and you cannot. I mean, it's difficult to place a time also when you're talking about more complex things. For instance, the first bacteria and other form of life. You cannot place you cannot put them in a place or in a time sometimes. So it's very complicated for that reason, I think.
Yes, so I'm just randomly seeing this here. So they found the age of this rock because they know the place from some geological settings, but for sure they make some determination. So because the first problem is some 20 years ago, they don't think they are fossils. Nowadays, we are almost sure that we are fossils because they haven't had the problem for the aging of the stone. So they are just questioning on if they are fossils. So I'm not an expert, but they trust about the age of the stone. Even this is not the only one. They found many. And many other fossils. This is not the best option that I found because there is one that I saw maybe some days ago. You see that the earliest occurrence of microbial fossils was in Australia. you see, already includes a filamentous form similar to trichomes. So we are not speaking about a simple life. So they are like cyanobacteria, so making photosynthesis. Also, they are aggregating in some filamentous form. So for us, it's so strange to think about this. So, but so we are speaking about I cannot know exactly which was the function inside these cells, but in any case, they were aggregating in some filaments. But the strange thing is that so we have fossils in these old, very old stones that looks like bacteria. Then for like another billion year, you have similar to bacteria, maybe with this filament not more. Then immediately the eukaryotic cell. That is the problem. Then animals.
Yeah.
So it's like a jump. But in any case, we have to think that the evolution, we imagine the evolution like a continuous process, but it's not like this. So the evolution, so the people studying the evolution from a theoretical point of view, they know that evolution goes by steps. So for a lot of time, it looks similar. Then you have a step because something triggering the change. So imagine, for example, this first bacteria, they found food. So as they are able to eat the food, they survive. Then the food finished, so they make some, for the pressure of the selection, they change. Like for when you have the formation of the oxygen, the same. So when the bacteria make the photosynthesis, produce the oxygen, at one time, the oxygen kills more or less almost everything. Then the species that have, so you have some pressures produced by, yeah, yes, exactly. That it's called punctuated equilibrium. So maybe this is the same, but this is still a big problem because nowadays we know, so not many billion years before, so 1 billion years before this, we have the formation of the moon. And then how this complex living system come here, so they spread directly. So many, yeah, okay, we have the pictures also. If you want, I can upload this paper, but I don't know if it is the most important. Just try to find this while you were speaking. So it's not the best one, but from here, maybe you can find some good references, hopefully.
Yeah.
But you see that the picture, they are like electron microscopic picture of actual organism. That is stunning. You see the aggregates in some forms. Okay. Okay, so you have other questions, or we will meet on Thursday? Okay, we'll upload this paper so you can have a look if you want. But I remember.
I found it.
Okay, good.
Yeah.
But the strange thing that I remember where it was a phosphorite, so a mineral having a lot of phosphorus inside. Okay, guys, so see you next time. Have a nice day.
Thank you very much.
Hi, Prof.
Thanks to you.
See you.
Bye. Bye.
Thank
Bye.
you.
Thank you.
Thank you. Bye.
Bye.